We are delightful to host three amazing early career researchers, Emmanuel, Roberta, and Leandros. And I will start with introducing our first speaker, which is Emmanuel Cazottes. So Emmanuel started his academic journey back in France, where in University of Paris, uh, he did his master's in molecular biology and genetics, and then in again City University of Paris, he did his PhD with Dr. Céline Moret on the evolution of X chromosome inactivation and regulatory non-coding RNA in primates. Uh, and then for his postdoc, he has now moved to University of British Columbia in Canada to the Carl de Boer Research, and have him as extensive remodeling of the net. Floor is yours. Emmanuel, can you hear me?
Thank you. Yes,
okay.
I can hear you. So thanks for the kind introduction. So today I'm going to present what I did during my PhD as I joined Carl de Boer's lab 5 months ago at the University of British Columbia in Vancouver, Canada. So during my PhD, I worked on the evolution. I worked on the evolution of X chromosome inactivation, and more particularly on the regulation of its main regulator, exist. So I guess most of you guys know that the mammalian genome is overly non-coding and can be and the major output of this non-coding genome is transcription. And among these RNA molecules, you can find long non-coding RNA genes that are matured RNA molecules that are longer than 200 nucleotides with little or no coding potential. So it's estimated that there is around between 20,000 to 100,000 long non-coding RNA genes in human. However, for now, we only have experimental evidence of described function for like 2,000 of them. When you look at the evolution of long non-coding RNA compared to protein encoding genes, you observe two drastic differences. So if protein encoding genes are quite fairly conserved throughout evolution, it's not the case of long non-coding RNA genes. So for instance, if you look at two species that have diverged for human roughly 30 million years ago, around 30% of long non-coding RNA genes are conserved, while 96% of protein coding genes are conserved. And if you look at two species that have diverged from human 160 million years ago, only 4% of the long non-coding RNA genes will be conserved with human, while 70% of protein coding genes will be conserved. So, and that's a direct consequence of the mode of evolution that is subjective to these two genomes. So the non-coding genomes is mostly subjective to genetic drift. And it means that mutations tend to neutrally impact the fitness of the host. And as a consequence, you have a poor nucleotide sequence conservation between species and a fast turnover of its constituents. While in the coding genome and a small fraction of the non-coding genome, it's subjected to natural selection. So here mutations tend to decrease or increase fitness, and as a consequence, sequences are more conserved between species. Nonetheless, maybe this rapid evolution of the non-coding genome could pose it as an incubator for genetic innovation. And this is a question I addressed in my PhD through the lens of X chromosome inactivation, which we believe is an interesting model to address such a question. So it's an epigenetic process of dosage compensation in mammals between female mammals and males. It is triggered during early embryonic development, by the expression of the long non-coding RNA exist that will coat the inactive X and enrich the inactive X for a very distinct chromatin landscape. So repressive histone modification, H3K27 trimethylation, H3K hub, and deplete the inactive X for active chromatin mark. X chromosome inactivation has been shown to be essential for the development and tissue homeostasis. However, surprisingly, we observed that we and others observed that XR strategies and its care regulators have diversified extensively during the mammalian evolution. So, for instance, in mouse, during the early embryonic development, you can observe two waves of X chromosome inactivation. The first wave is happening very shortly after zygotic genome activation and is imprinting, meaning that it's going to be the paternal copy, the paternal X that will be systematically inactivating following the upregulation of exist. At the blastocyst stage, the exist will be repressed from the paternal X, and random inactivation will follow upon implantation. So, however, in human, you have a different scenario where both X chromosome express X chromosome inactivation without triggering X chromosome inactivation, which is only resolved later on during upon implantation. So here, what you can see, if you have two strategies, first in mouse, you must upregulate one exist on one X only to induce XCI, while in human, you must downregulate exist on one X only to induce random XCI. And we believe that those differences are tied to how exist is regulated within its homing locus on the X chromosome. So, and more specifically by two neighboring long non-coding RNA genes, namely GPX and FTX. So in mouse, GPX and FTX are required to upregulate exist, and they do so by first. So GPX RNA molecule is required to upregulate exist, and the transcription of the FTX locus is required to upregulate exist in mouse. However, in human, if both genes are conserved, they have different functions. So in mouse, a former PhD student of the lab, Olga Rospopov, have shown that actually this is a GPX transcription that is required to upregulate exist in human, and FTX does not have any function related to X chromosome inactivation. So for my PhD project, we sought to investigate the evolution of this regulation by neighboring long non-coding RNA, but to do it like on a very short evolutionary timescale to have the resolution to pinpoint the differences that would have happened during evolution. And we focused on two primates that are closely related to human. So the marmosets that have diverged from human 30 to 35 million years ago, and the rhesus macaques that have diverged from humans for 30 to 35 million years ago. So I first started by looking at the X chromosome inactivation status in stem cells derived from those two species. So in human embryonic stem cells, they have established X chromosome inactivation and resemble a transcriptomic state of the post-implantation epiblast. So what I did is I did an RNA fish, so staining the RNA within the cells with fluorescent probes for exist and two X-linked genes, HRX and POLY1, that are known to be subjected to X chromosome inactivation. And in most of the cell population, I could detect one cloud of exist with one pinpoint of HRX and one pinpoint of POLY1, indicating that X chromosome inactivation has been undergone in those cells. And we also did RNA-seq from the cells to look at the expression of polymorphism on the X and two autosomes. So for two autosomes, you can detect biallelic expression, while on the X, you only detect monoallelic expression, indicating that those cells have undergone a full and complete X chromosome inactivation. I did the same thing in marmoset, RNA fish again for exist and ATRX. And in the most of the cell population, you have one cloud of exist and a pinpoint of ATRX away from the exist cloud, indicating that those cells have undergone X chromosome inactivation. We did further analysis to show that those cells match to the same developmental stages, the post-implantation epiblast. So our prime protein stem cells map to a similar developmental stage in vivo and are amenable between species comparison. So I first started with the macaque, and I first mapped the expression pattern of exist, GPX, and FTX in those cells. So all of the three genes are co-upregulated throughout the female rhesus pre-implantation embryogenesis. And I did RNA fish for GPX, FTF, and exist to get a sense of their expression patterns. And what I could see is I get only one pinpoint for FTX away from the exist cloud, and together with that colocalized with GPX pinpoint, but also in almost half of the cell population, I also have one pinpoint near the exist cloud. So it says that FTX is subjected to X chromosome inactivation while GPX partially escapes in X inactivation. So it's expressed from the inactive X. And I wanted to know, do these two guys have any actually regulating exist? So to address this question, what I did is I performed a CRISPR knockout of the whole GPX and FTX locus in rhesus embryonic stem cells, and I derived two kinds of genotypes. I focused on heterozygous genotypes because I reasoned that having heterozygotes could question whether when the deletion is happening on the active X, could the locus have a trans effect on exist regulation, or when it's happening on the inactive X, does locus have a cis effect on exist regulation? And this is what you're showing here. When the deletion is happening on the XA, you completely lose FTX expression that is expected because it's only expressed from the active X. When the deletion is on the XI, you don't have a significant downregulation of GPX because most of its expression comes from the active X. So now what about exist? And what I could see is exist expression was downregulated, significantly downregulated only when the deletion was sitting on the inactive X. So it indicates that GPX FTF locus have a cis regulator of exist in macaque embryonic stem cells. I then wanted to know what is the impact of this such regulation on the control of exist RNA levels. So to modulate exist expression level, or does it play a role actually on the probability of the activation of the promoter? And I leverage the RNA fish data we have and the analysis to show and expression analysis to show that indeed, while there is a decrease of exist expression, you also have a decrease of the volume of the exist cloud in the nucleus. And I follow, but what was quite surprising to us is while you delete the locus, you still have a consistent cloud of exist in the nucleus. And so it seems that the GPX FTX plays a role on controlling exist levels, but not its probability of activation. So now I wanted to know who does the job in regulating exist in macaque stem cells, and I use the CRISPR system to repress GPX directly. So upon, this is an inducible system, upon induction of the transgene, I get a very efficient repression of GPX. As you can see, we like here by as measured by RT-qPCR, but by RNA fish, I do not detect GPX after repression of the transcript. So armed with that, I could show that when GPX is repressed, I observe a significant downregulation of exist, a reduction of the exist cloud, but I still see an exist cloud in every cell. So to us, it indicates that GPX phenocopies the deletion of the whole locus and is very likely to do the job in regulating exist. So And it's actually quite a bit of it. When you compare to the species where, for instance, in mouse, GPX is required for both the activation, is required for exist promoter activation and modulation of the RNA levels. And in human, it is required also for the activation and modulation of the RNA levels. However, what we observed in the macaque, it's GPX only modulates exist RNA levels. So I wanted then to look at another species that is the marmoset to get a sense of what is the ancestral state in primates. And and for the second time, I'm sorry, I'm gonna skip to the conclusion because I'm a bit late. And so, sorry, I'm not showing all the data, but briefly, to summarize what I'm trying to show today is I could show that in all species, GPX is a conserved regulator of exist, yet through different mechanism and mode of action. Particularly in macaque, where only GPX is only modulating exist expression levels, while in the marmoset and humans, it's actually also modulating the probability of the promoter, the activation of the promoter. And which was actually surprising is like this, if this change is conserved, it's only by sheer random chance and not the act of natural selection. So that was, but I only presented a small fraction of this work, and there is actually more, where in the macaque, we also describe that those changes are associated, with an evolution of the 3D chromatin landscape and the addition of an enhancer that works together with GPX in controlling exist. So if you want to know more, feel free to check out the preprint that is on bioRxiv. And with that being said, I'm gonna acknowledge people from the Rouge Lab. So Céline, that has been my PhD director, Charbel, and Gael, who helped me a lot with some of the omics and analysis, and our collaborator on the pluripotency in primates, Capture-IC, and phylogenetic analysis. With that being said, thank you all for your attention, and I'll be happy to take questions.
Emmanuel, thank you for the great talk. If anybody has a question, either you can raise your hand and I can unmute you and you can ask your question, or you can write in the Q&A box. You know the drill.
Can I jump in with a question?
Yes, Jackson, go, jump in.
Yeah, that was really fascinating. And you tease it here in the conclusion. I wanted to ask about whether endogenous retroviral elements or differential like invasion of the X chromosome by viral elements would be responsible for this sort of evolutionary difference.
So I think it's a bit like retroviral evolution, like retroviral invasion, sorry, is quite pervasive, and it's a bit hard to pinpoint. For instance, if you look at the structure of GPX throughout those three primates, they are completely divergent. There is no constraint there was no selective constraint to maintain GPX splicing structure. And even in the macaque, it seems that GPX is not spliced. And it's mostly those divergences are mostly due to the insertion of transposable elements. So is it a hotspot of insertion events because there is no selective pressure and we see that and the insertions are maintained? Or is it also insertion are actually a driver of this evolution? To me, is not known.
Thanks.
Sorry. I was saying that we are out of time. For more Q&A questions, I think Emmanuel can respond them in the Q&A box, typing the answers. And now I will hand the mic to Sky, which will introduce our next speaker.